Hello one and all gathered here. I am Ms. P. Amirtha, lecturer, Department of Biotechnology, Sita Lakshmi Ramaswamy College, Trichy. So today I am going to teach you about cell division, especially mitosis. So cell divisions have classified into two divisions. One is mitosis and another one is meiosis. So cell division happens when a parent cell divides into two or more cells called daughter cells. Cell division usually occurs as a part of a larger cell cycle. All cells reproduce by splitting into two, where each parent cell gives rise to the similar species of two daughter cells. So, by the help of this diagram, we can see the variety of phases involved in cell division. First A part is interphase. In the interphase, the centrosomes are separated into two categories into a nucleus. Next, early prophase, late prophase and metaphase. These are the three major phases which involved in a process. So spindle poles are just attached and it was separated with the help of early prophase system. Next, the kinetochore were attached with the sister chromatids and it forms the metaphase process. And anaphase, telophase, interphase are the next major parts involved in a cell division. So, in the anaphase, the entire centrosomes will be divided into two daughter cells as a telophase and interphase. At last interphase, we can see the two similar daughter cells as similar as a parent cells. So these newly formed daughter cells could themselves divide and grow, giving rise to a new cell population that is formed by the division and growth of a single parental cell and its a descendant. In other words, such cycles of growth and divisions also a single cell to form a structure consisting of millions of cells. So, what are the types of cell divisions involved and what is the difference between the mitosis and meiosis? We can see further. There are two distinct types of cell division as I had already told, out of which the first one is vegetative division, wherein each daughter cells duplicate the parent cell called mitosis, and the second process is called meiosis, which divide into a four haploid daughter cells. The two daughter cells will be called as the mitosis and four will be called as the meiosis. So mitosis is defined as the process of cell used to make exact replica of themselves. Mitosis is observed in almost all the body cells including eyes, skin, hair and muscle cells. In the process of meiosis, this type of cell division, particular in sperm or egg cells, are produced instead of identical daughter cells as in the mitosis. So from this diagram you can see how the G1 phase, S phase, G2 phase and M phase will be mitotic in the process of resting. At the starting stage, the G0 stage will be circulated into a G1 phase. In the interphase, the entire process will be moved to the S phase, G phase and metaphase. So, from this diagram, we are just explaining the further mitosis and meiosis diagram. So, here I had shown you the prophase, pro-metaphase, metaphase, anaphase, telophase. The difference between the phases will be clarified as the images here. Next, mitosis. Now, we are going to see the process involved in mitosis based on the phases of four categories. Division phase or M phase or mitotic phase. This will be known as the varieties of name. Lasts only about 1 hour and 24-hour duration of cell cycle of a human cell. The M phase represents the phase when the actual cell division or mitosis occurs. In animals, mitotic cell division is restricted or only seen in diploid somatic cells except in some social insects. Against this, the plants can show mitotic division in both haploid and diploid cells. The M phase usually starts with the nuclear division. Corresponding to the separation of daughter chromosomes, the karyokinesis. Karyokinesis shows they usually end with the divisions of cytoplasm, that is cytokinesis. This will start from the karyokinesis process and ends with the cytokinesis. So mitosis has been divided into the four major stages of nuclear division: prophase, metaphase, anaphase and telophase. So let us see the four divisions of phases with a clear explanation. So first one is prophase. Prophase, which is the first stage of karyokinesis of mitosis, followed the S and G2 phase of interphase program. In the S and G2 phase, the new DNA molecules will form and not distincted but intertwined into the daughter cells. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material become untangled during the process of chromatin condensation. The centrioles is the particular round part of a chromosome, that is a centromere, which has undergone duplication during S phase of interphase. Now it's begin to move towards the opposite poles of a cell. The second position, that is the second stage, is called metaphase. The complete disintegration of nuclear envelope marks the start of a metaphase. By this stage, the condensation of chromosomes is completed and they move to the observed clearly under the microscope. Then the stage which morphology of chromosomes are most easily studied and we can see through the microscope process. At this stage, the metaphase chromosome is made up of two sister chromatids, that is a daughter cell developed part, which are held together by the centromere. Small disc-shaped structure at the surface of centromeres are called kinetochores. These structures serve as the sites of attachment of spindle fibers. Spindle fibers are also formed by the microtubules to the centrosomes that are moved into the position at the center of the cell. Centromere lies at equator and arm of the chromosomes remain directed towards the poles. And the third phase is called anaphase. Anaphase is the one of the important phase in the mitotic division. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously and the two daughter chromatids, now it's referred to as the daughter chromosomes of the future daughter nuclei, began their migration towards the opposite poles of each other. As each chromosomes move away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at the leading edge, with the arm of the chromosomes that trailing behind the process. So the number of chromosomes will be doubled in the cell. As the parent cell have a single centromere and the daughter cells will be segregated into two. So it will be doubled in the second phase of the mitotic division. So the two new daughter chromosomes begin moving towards opposite end and cell as their spindle fibers shortened into it. Next, the final stage is called telophase. This phase is also called as the reverse prophase. As we have already learned about the prophase, the reverse prophase is called as telophase. At the beginning of the final stage of karyokinesis, that is a telophase, the centrosomes that have reached their respective poles and lost their individuality. So at the individual chromosomes can no longer be seen and chromatin materials tends to collect at each of the two poles. So in this image we can see the first part, the first nucleus part, the two chromosomes are separated each other and the replication occurs in that second part and the chromosomes will be developed into that. And next category, the spindle fibers are connected each other from the corner of the part with the help of centromeres. And at the process of mitosis, it will be divided into a 4 haploid cells and at last the two nuclear daughter cells will be separated into a diploid cells. So mitosis is a process of duplication or reproduction during which one cell gives rise to the two genetically identical daughter cells. Strictly applied, the term mitosis is used to describe the duplication and distribution of chromosomes. Thank you.